Hello, and welcome to this lecture on human embryonic stem cells created from adult somatic cell nuclear DNA. So to start out with this series of lectures on research ethics, we're going to talk a little bit about the science, about the problem that the science is trying to be used as a tool to solve, as well as some ethical issues associated with it. And so what we're looking at in this lecture is going to be working with embryonic stem cells. And so to start with, we're going to look at the creation of an embryonic stem cell lines. Normally, this is going to occur using techniques very, very similar to in vitro fertilization. And basically what's going to happen is within a laboratory setting, we're going to bring together donated sperm with donated eggs to allow for, as the slide indicates, in vitro fertilization. And so the egg and the sperm are going to come together, fertilization is going to occur, and still within the laboratory setting, very quickly, that fertilized egg is going to go through the process of cleavage, rapid cell development without a whole lot of growth, where we're going to have kind of a ball of cells that are going to be forming. And then ultimately that ball of cell within the first week, again, we're looking at human embryonic stem cells, that ball of cells is going to develop into a blastocyst. For those of you kind of remembering your developmental biology from maybe Biology 102 or from another course, a blastocyst is a hollow ball of cells. And so we've got, in essence, the blastula here, which would have been the term you used in Biology 102. So we've got a layer of cells kind of around the outside. We've got a blastocele. We've got a hollow cavity on the inside. And that's the characteristic of a blastula or blastocyst. Now, what's a little bit different with human embryos than in other types of embryos is that what we're going to be looking at is that in the production of this blastocyst, we're going to have two very distinctly different sets of cells. We're going to have cells of the trophoectoderm kind of around the outside, and these are still cells formed from the fertilized egg. So they're, the new organism's DNA is going to be found within them. But these trophoectoderm cells aren't going to be involved with production of the embryo, which will become the fetus, which will become the new organism. The trophoectoderm cells are going to interact with cells within the maternal environment, within the uterus, to form the placenta to support and nourishing the developing embryo and fetus. So what we're going to look at is focusing in on these cells along the inside. These cells here within the inner cell mass are going to be the cells that ultimately are going to develop into the embryo. They're going to develop into the fetus. They're going to develop into the new organism. And so at this very early stage of development, this inner cell mass is going to be a cluster of cells which are going to be capable of becoming all of the cells of the mature human that we're going to be looking at. So these cells, these inner cell mass cells, then have the potential to develop into any of the types of cells within the body. What's going to happen when we're creating an embryonic cell line, embryonic stem cell line, is we are going to isolate these cells within the inner cell mass, and we're going to grow them in a laboratory setting. Grow them on a petri dish, as we're indicating here, or some other type of culture setting. And what we're going to see is that these embryonic stem cells, these cells that are derived from the inner cell mass, under the proper circumstances, are going to continue to divide. And they're going to stay undifferentiated. So they're going to stay stem cells. They're going to be able to generate lots and lots and lots of these stem cells, which can be studied and used in a variety of ways. Now, what we're interested in is the fact that we can take these stem cells, these embryonic stem cells, these cells that are isolated from that inner cell mass. Again, a slightly different drawing associated with this. So we've got the blastula here, the blastocyst, the blastocele, the cavity to the inside, the trophoectoderm cells kind of around the outside, and then focus then on these cells within the inner cell mass. We're going to isolate those cells in the inner cell mass. We're going to grow them in the proper conditions within a culture setting, on a petri dish as it's illustrated here. And if you keep them under the proper conditions, they're going to stay undifferentiated. They're going to stay unspecialized. And when they're unspecialized and undifferentiated, we can get them to divide over and over again. Ultimately, though, if we continue to allow them to divide, to come in contact with one another, start producing factors that are going to allow the cells to interact with one another, these embryonic stem cells will often form little embryoid bodies. And these embryoid bodies kind of show the potential of the embryonic stem cells. These embryoid bodies have the potential to become any of the 3 germ layers or any of the cells that are derived from the 3 germ layers. And so again, kind of reviewing your developmental biology, the endoderm is going to be normally found kind of lining the gut, as well as some of the accessory organs of the digestive system or the lungs. And so that's the endoderm. The cells of an ectodermal lineage are going to normally develop into the skin, but they're also going to develop into the nerve cord. And so they're going to develop into the spinal cord and the brain, as well as some specialized nervous system structures. And then in between the endoderm and the ectoderm are going to be the mesoderm. The mesoderm-derived cells are going to be basically everything inside. So it's going to include the blood, the muscles, bone, cartilage, many of the organs that we're going to see within the body. And so when we're studying embryonic stem cells and we want to learn how to use these embryonic stem cells, what scientists are going to do is focus in on differentiation factors. And these differentiation factors are going to be factors that are going to allow these embryonic stem cells to start to become specialized, to start to become differentiated. And so what's going to happen is that when these embryonic stem cells are treated with differentiation factors, they're going to stop dividing as much as they were before, and they're going to start to become specialized. You're going to start down one of these paths, as illustrated over here, and basically come into kind of a germ line, and then within the germ line, specialized cells are going to develop out of that germ line. Normally in development, we're going to see these differentiation factors as being like growth factors that are secreted, in the developing embryo, maybe provided by cells kind of in the neighborhood. There can be also morphogenetic factors that again impact on these cells and push them into a specialization pathway. What we've learned by studying these embryonic stem cells in culture is that there are a variety of other things that can either naturally or artificially push them into a specific phenotype, a specific type of developing specialized cells. And this includes using different feeder cell layers, so basically growing these embryonic stem cells on different types of cells and cultures, and those cell interactions and the factors that are released by the cells within that culture condition can push the cells into a specific phenotype. And it's also been learned that just simple extracellular matrix molecules like laminin or fibronectin, putting those proteins, glycoproteins, onto the plate of a tissue culture dish will start to cause some of these embryonic stem cells to differentiate. And so basically, if you're studying these stem cells, the idea is that you start out with these undifferentiated cells, lots and lots of them because they're capable of dividing over and over again. But ultimately, we want to take some of these cells, not all of them, because we want to continue to have a continuous renewable supply of these cells. We're going to take some of these cells and expose them to differentiation factors and look at that differential gene expression. Those differentiation factors are going to activate sets of genes that are going to push these cells into these kind of developmental phenotypes, these specializations that we're going to be looking at. So the question is, why are we interested in studying embryonic stem cells? Why is it important? And so we're going to take a little bit of a step back from the science of stem cells and look at basically transplants of cells or transplants of organs for individuals. Now, a very classic example of what in reality is stem cell, not embryonic stem cell, but stem cell transplantation, is the idea that you can do bone marrow transplants in patients. So if you've got a patient here, a recipient here on this diagram, this would be a patient that has potentially some type of bone cancer that affects their hematopoietic cells. Hematopoietic cells are going to be the cells that, again, are found in the bone marrow that are going to continue to divide throughout an individual's lifetime. And some of those cells over time are going to develop into blood cells, either red blood cells or white blood cells. When an individual gets one of these blood cancers or a bone marrow cancer, what happens is that these diseased hematopoietic cells are going to start to kind of divide kind of in an uncontrolled fashion, or start to differentiate in an uncontrolled fashion, and you end up with something like leukemia. And so the idea is that we want to get rid of those cancerous or abnormal hematopoietic cells, these diseased hematopoietic cells in the patient. And so basically we treat them with either radiation or an anti-cancer drug to kill off all of those cells. But while that may cure the leukemia, cure the blood cancer, it now puts this individual at extreme vulnerability because they can't replace their blood cells, either red blood cells or white blood cells. And so we need to replenish or replace those damaged or diseased hematopoietic cells that we killed off. And so what's going to happen is we're going to take a bone marrow sample from a healthy donor, collect these hematopoietic cells, these stem cells, and put them into the recipient with the idea that these cells that are coming from the healthy donor injected in the recipient will become established within the bone marrow, will continue to divide, and will continue to produce blood cells for that recipient. But what we know is that we need to have a good match between the donor and the recipient. And the science behind that is that there are going to be protein markers on the surface of our cells. The most common one, the ones that we focus in on when we're looking at matching a donor to a patient, tend to be the HLA proteins. The HLA is a human leukocyte antigen. These are protein markers that are found on the majority of the cells within our body. And our immune system uses these HLA markers to identify self versus non-self. So very early on, these immune system cells are going to learn what HLA molecules are normally found within the body. And they're going to basically, in a process almost like learning, but we're going to kill off those immune system cells that would attack our body cells and prevent an autoimmune disease, in essence. And leave behind those cells that are capable of recognizing HLA antigens that are different from what are normally found within the body. And normally that would indicate, some type of disease state or pathogen that's coming in. So the idea when you do some type of cell transplant or organ transplant is that you want to match these HLA antigens from the donor to the recipient as well as possible to minimize what would be graft versus host disease. Now in graft versus host disease, we're going to end up with rejection of the donor tissue. In other organs, if we're looking at like a heart transplant or a liver transplant where we haven't affected the immune system cells like we're doing in this example here, we could take a heart out of a donor, put it into a recipient, and their immune system, if we don't have a good HLA match, will recognize those cells as being different from normal, what's normally found within the body, and start to mount an immune response to it and really kill off those cells relatively quickly. And that's bad because that would be not beneficial to our patient or recipient who probably had heart disease to start with, now had a replacement heart put in, but the immune system is attacking it. The other thing that can occur instead of rejection of the donor tissue, especially an example where we're moving hematopoietic cells from a donor to a recipient, is the fact that these donor cells are going to develop into immune system cells. And if we don't have a good HLA match, what can happen is the immune system that's created by the transplantation of these donor cells into the recipient can actually start to attack the host cells, start to kill them off, because they're going to attack what they perceive as being foreign or diseased. And in this case, those foreign or diseased cells would be the normal body cells within this recipient. And so without a good HLA match, we're going to have this graft versus host disease and a lot of complications associated with it. With a good HLA match, we're going to have less of a risk for that. But in many cases, what happens is we still need to treat these patients, these recipients, with immunosuppressants, which will turn down the immune system response and minimize the likelihood that these transplanted tissues will either elicit or cause a death of cells, this graft versus host disease. The reason this is especially challenging is that patients that are on immunosuppressants are susceptible to future pathogens. With these future pathogens that they may be exposed to, with a suppressed immune system, they're going to be more likely to get a disease in the future. And so an ideal way of doing a stem cell transplant or any type of organ transplant is to use cells that match up, automatically match up exactly with the recipient. And so, if we're going to move an organ from, a donor to a recipient, you want them to be, genetically identical. You want them to be identical twins. But that, isn't an option for most people. And so an example of this would be an autologous stem cell transplant, to basically take cells from a patient, process them in some way, and then put them back into the same patient. Because the patient is receiving their own cells, they would potentially then not have that risk of graft versus host disease. And so this is a slightly altered version of a diagram from the cancer NIH Cancer Institute. And so what we've got, and again, this is modified, this had, another patient in it when we started, a donor, and we brought over the patient here. So you collect stem cells from the patient. Now it's hooked up to an apheresis machine, which basically separates, blood from stem cells from other things. So we can take out the stem cells, potentially process them in some way, or isolate and grow them. Treat them with, once we've got these cells that have been isolated and grown, take the patient, expose them to something that's going to kill off the cancerous cells. In this form, we're looking at chemotherapy, destroying those cells. And then ultimately, the patient, after these disease cells have been killed off, are going to receive their own stem cells or cells that are matched to them because they're coming directly from them. And again, this is an autologous stem cell transplant. As opposed to what we were talking about before, an allogenic. An allogenic is coming from another individual, and you have to worry about the HLA match because of the graft versus host disease. So ideally, we would be able to kind of find cells from a patient, modify them in some way, and then use them in the patient to create a situation where we're treating some type of disease process, some type of damage to their normal body cells. And so what this is a form of cell replacement therapy. So examples of human diseases where we could use this cell replacement therapy, assuming that the cells are available for us, would include things like nervous system diseases like Alzheimer's disease and Parkinson's disease, where you would have kind of death of cells. And by taking and creating stem cells that become nerve cells, you could potentially kind of alleviate or prevent the effects that we're seeing with these diseases. Type 1 diabetes, where you've got a loss of the endocrine secreting cells within the pancreas that are normally secreting insulin. If we've got stem cells that are kind of pushed in the right direction, like the diagram is showing here, that can produce insulin, we can now inject them into the patient and potentially cure type 1 diabetes. Other things, a whole variety of examples here, heart disease and liver disease. The idea that we could take cardiac myocytes or hepatocytes for the liver, inject them into a damaged region and repair those organs. This has great potential but it has great risk for it if we can't do that HLA match and have the ability to minimize the graft versus host disease. Ultimately, at some point in the future, it may be possible to do tissue engineering where we take these stem cells and, as opposed to injecting in individual cells, build replacement artificial organs that can then be put in, so that we're using the patient's own cells to build a replacement organ instead of waiting on a transplant list, waiting for somebody to have an organ that's available for you, and then hoping that their HLA match will be sufficient to allow you to use that in your body without rejection. So there's great potential if we can take cells from an individual, from the skin cells in some cases, alter them or reprogram them to become stem cell-like, and then push them in the right direction so they develop into the phenotype. In this case, the phenotype would be endocrine secreting cells, glucose-sensitive, insulin-secreting cells that we can inject in and replace the function that's been lost in type 1 diabetes. There are great limitations in the ability to reprogram adult cells, and so we really need to come up with a way in which we can generate stem cells. We can generate these very unspecialized, undifferentiated cells that we can push in the proper direction. Another approach that we could take is this concept of somatic cell nuclear transfer. In somatic cell nuclear transfer, what we're working with is taking donor cells from an individual, isolating their nucleus. Within this nucleus, all the genetic material, so it's an exact genetic match for the donor. And in this process, we're going to take an egg cell, an ova, enucleate it, so remove the nucleus, but this egg cell has all of the protein machinery and all of the signals and everything necessary for development if it was to be fertilized. But instead of taking this egg, which would normally have a nucleus, and bringing sperm cells in and doing in vitro fertilization, by wiping out this nucleus, we can bring it up here, take our donor nucleus, put it into this egg cell, and now we're going to have an egg cell with a diploid, 2 sets of chromosomes within this donor nucleus. The sets of chromosomes are going to be genetically identical to the donor. We're going to put them in here, activated as represented by this electric shock here. What we're going to have is going to be the equivalent of a fertilized egg. We've got a diploid nucleus within it, not diploid because we got the egg nucleus coming together with the sperm nucleus, but diploid because we took the genetic information from a donor and put it directly into this egg cell. If this is done properly and a whole lot of luck and a lot of other things that we don't necessarily understand, we're going to have this diploid cell, exact clone of the donor, with all of the protein machinery in it, and it ultimately is going to develop into a blastula, develop into an embryo, and then we're going to take that embryo and plant it into a surrogate mother. Like if we were going to do in vitro fertilization in a human setting, we would do in vitro fertilization, allow the embryo to develop to the appropriate stage, and then implant it into either the uterus of the mother or into the uterus of a surrogate who would be allowing the child to develop within their womb. Ultimately, again, assuming everything works along each one of these steps, we're going to see the birth of an offspring here. But this offspring then is going to be an identical clone to the donor up here, because that nucleus with all of its genetic material was what went into the egg, and when the cells divided, that nucleus is going to divide so that all of the cells of the developing embryo that's implanted into the uterus of the surrogate mother, all of the cells here are going to be genetically identical to the sheep. And so that's the process of somatic cell nuclear transfer. And this was first demonstrated in a mammal in 1996 when scientists were able to create Dolly the sheep. And so this is incredibly exciting. It was a very difficult process. There were a lot of challenges to do that, but it has great potential because ideally you could take cells from some type of patient up here, isolate their nucleus, combine it, under proper circumstances with an enucleated egg and generate lots of cells. So if we take a look at this, what we've got here are two papers that came out in 2004 and 2005 that reported that they were able to successfully use the same technique, somatic cell nuclear transfer, to create embryonic stem cell lines. And this occurred in 2004 and 2005. And so this slide is very similar to what we had on this previous slide, where we looked at the creation of Dolly the sheep. So, we've got a donor here. We're going to take somatic cells, body cells, isolate the nucleus. We're going to have human ova, in this case, human egg cells. We're going to get rid of the nucleus to form an enucleated egg here. And we're going to take that somatic nucleus, diploid nucleus, 2 sets of chromosomes, genetically identical to the donor, put them into the enucleated egg, activate it, and then, like in the process of in vitro fertilization now, we're going to allow this activated egg, not fertilized, so it's not truly in vitro fertilization, but we, brought it, the somatic cell nucleus into the egg cell. So we do have a diploid kind of activated egg here. We're going to see development to the blastula stage. So we've got the trophoectoderm cells around the outside. We've got the inner cell mass here to the inside. If we're going to do in vitro fertilization, the equivalent of that, making a reproductive clone, a baby that's a clone of the donor here, we would just take this, allow it to develop, and then implant it into a surrogate mother. But what we're looking at in this series of studies is taking this, isolating the cells of this inner cell mass, and creating embryonic stem cells, like the embryonic stem cells we started talking about at the beginning of this lecture. And so these embryonic stem cells, again, very undifferentiated, very unspecialized, capable of dividing to produce lots and lots of copies of them. And we can take some of those cells and push them into a specific phenotype. So this is incredibly exciting. In 2004, they represented doing it with 1 cell line from a test subject. In 2005, they reported doing 11 embryonic cell lines from donors, and they also reported incredible efficiency. They were able to, according to the papers, generate 11 embryonic stem cell lines from 185 eggs, and this is incredibly efficient, indicating that potentially they, figured out the problems we had before. When we looked at creating Dolly the sheep, they ended up having to try this, almost 300 times to get a successful offspring being generated. A lot of the studies that have attempted to do this, are doing it hundreds or thousands of times to have one successful category or one successful lineage established. In this case, 11 lines at 185 cells is basically 1 line for about every 10 cells, 10 eggs that they're working with. Incredibly, incredibly efficient. This is very important work, as illustrated by the fact that the authors of this science study have received US patents for this technique and modifications of their technique in 2003, 7, 10, 11, 12, 14, and 16. So they're continuing to work on this, continuing to fine tune it, and continuing to receive patents associated with it. So this is incredible. This is the idea that we can take basically cells from a patient, go through this process and generate lots and lots of potentially therapeutic cells that we can use to treat a disease state in this individual. And because they have the nucleus from their own somatic cells, all of these cells are going to be genetically identical to the patient, to the donor that we're working with. So this minimizes greatly the risk that we'll inject these cells into the patient and have graft versus host disease or a rejection of these implanted tissues. So this is incredible. This is an amazing achievement. If we take a look at the main scientist leading this is Dr. Woo-Suk Hwang in Korea, South Korea. He became a national hero, a scientific leader, With the publication of these papers, government agencies, private research foundations were throwing money at him to support his work. So he received lots and lots of grant support and other sources of funds. He was recognized by Time magazine in 2004 as one of the people who mattered. Scientific American identified him as the research leader of the year in 2005. They established a World Stem Cell Hub to kind of launch this new world, this new kind of application of stem cell technology in 2005, and made him the head of it. There was talk, within a year of this paper coming out, that he was a candidate for the Nobel Prize in Medicine. So this is incredibly, incredibly significant. This was a huge accomplishment. However, over time, there were a number of ethical issues that gradually emerged associated with these studies. Some of them, kind of starting out with, were the questionable source of the eggs that they were working with. Again, recognize that we're using human eggs that would be enucleated, that would receive that somatic nucleus. And so it came out very early on that some of the eggs were coming from researchers in the lab, students in the lab, technicians in the lab, other people working in the lab. And that is a little bit ethically concerning because there's a risk of coercion. There also came out that they were making payments to, in essence, purchase the eggs from donors, which was occurring in violation of the South Korean bioethic law. So that raised some issues. Along the same time, as they were looking at the source of the eggs, they were able to determine there was insufficient informed consent. And so in order for a woman to donate eggs in this way, they would have to go through a kind of a hormone treatment so that they're producing lots and lots of eggs, as opposed to the single egg in an individual cycle, producing lots of eggs in a cycle so they can collect many of them with a single surgery. There's some risks associated with it, some things that the donors need to be aware of, and those weren't necessarily made clear to the people that were donating it. More significantly is that some of the co-authors started to raise questions about the actual findings. One of the co-authors said, pretty much almost as soon as the 2005 paper came out, that 9 of the 11 cell lines that were claimed in the study never existed, and 2 of the other lines, 2 of the remaining lines, were questionable origin. So scientific groups started to look at these cell lines, and they determined that the DNA from the cell lines didn't match the DNA of the donors. Again, if this technique was done as they described, these cell lines would be genetically identical to the donors because they would in essence be clones, specialized cells that are cloned from that donor. As they looked into it further, they indicated that some of the images within the paper were mislabeled, or they came from other cell lines from other sources. And so lots of issues with it there. It was also claimed that they falsified some of the data. They were basically using a lot more eggs than claimed. So the process wasn't efficient as it claimed. Instead of 1 out of 10, they were probably still doing the 1 out of several hundred to be able to generate a successful line, even if those lines, as raised questionable, appear with the potentially fabricated data. So it basically, wasn't necessarily accurate at what was going on. So multiple investigations of the work ensued, and again, these papers came out in 2004 and 2005. Very quickly, within January of 2006, the Seoul National University, where Dr. Hwang worked, announced that the stem cell lines had been fabricated, that they'd been kind of made up, and called for the retraction of both the 2004 and 2005 papers. Within a couple months of that, Dr. Hwang lost his stem cell research license. He was the only one that had that license in Korea. And because of the ethical issues and the concerns based on what he was doing, he lost that and was ultimately fired from Seoul National University, where he was a professor. Outside of the research world, within a couple of years, in October 2009, he was found guilty of embezzlement of some of the research funds, basically misdirection of funds, using funds for personal use, or at least uses different from what the people that were providing the money had intended it to be, as well as ethics violations involved with collecting the eggs in kind of unethical ways. What's interesting is that the court did not find him guilty of research fraud, even though the research community pretty much accepted that this work was fabricated and falsified, and ultimately retracted. Other people caught up in this study. Senior co-author Gerald Schatten from the University of Pittsburgh was loosely affiliated with the studies, had been involved with some of the other studies being done within the lab. His presence added international credibility to the studies, but in reality, he was not directly involved or directly knowledgeable about what was happening. And an investigation into his efforts determined that he wasn't guilty of research misconduct, but it was determined that he was guilty of research misbehavior. And this is kind of a new way of describing things. Basically, a report said that he was shirking his responsibility as senior author of the 2005 paper, and in that role as senior author, he should have had a better understanding about what was going on in the lab, and he should have ensured the veracity or the accuracy of the data and the results that were being presented. So that whole kind of 2004-2005 papers kind of raised lots of expectations that came dashing down. What's interesting is that in August 2005, kind of in the midst of all of this going on, this same group, we got Dr. Wang and Dr. Schatten here in this photograph. They reported in Nature, a different scientific journal, the cloning of an adult dog using the somatic cell nuclear transfer technique. And so basically, the dog, this Afghan hound here named Snuppy, was created in the way that we talked about before. We took, or they took, somatic cells from an adult donor, took the nucleus, put it into an enucleated dog ova, allowed it to develop, put it into a surrogate mother, and Snuppy was born. They were able, with all of this stuff going on, all the investigations for the 2004 and 2005 paper, it was independently verified doing genetic testing of Snuppy that Snuppy was a true clone of the adult dog donor. And so this paper, even though all those other things were going on, this paper appears to be accurate. They were able to clone an adult dog and create a new puppy called Snuppy. So that, they were doing, at least some of these techniques properly and developing it and advancing what was known within the field. But the 2004-2005 controversy really kind of raised a red flag and limited the ability of Dr. Wang to be able to work within this field. So the question is, what has Dr. Wang been doing lately? Dr. Wang has left academia and has started his own private biotech firm. And what's interesting is in the time that he's done that, he's done a whole lot of other things. He's started out collaborations with groups in this case, we're looking at it in Russia and other organizations where they're going to attempt to do the somatic nuclear transfer to clone a variety of other endangered species. Very early on, about 2013, there's a National Geographic special about his efforts to clone a woolly mammoth. And the major source of funding for his firm is the fact that he will do dog cloning. And so if you have a dog that you really like and you would like to have another one that's exactly the same, they're able to clone it. They've actually developed a technique that if they get cells from a dead dog within a set period of time of the dog dying, they can take the DNA from that dead dog and clone a new puppy of it. That group has done about 700 puppy clones since 2005 at about $100,000 each. So very, very pricey, but you get a genetically identical puppy to what you had. It doesn't say anything about the environment and the raising, the training, and all of that that's going to be going on with the dog that may kind of impact on the kind of puppy you get and the behavior of the puppy that you're working with. So lots of things to consider in this study. Some potential questions to consider as you're thinking about this. Does the scientific misconduct associated with the retracted 2004 and 2005 science papers diminish the significant accomplishments Dr. Huang has made in cloning other animals? I haven't shown you this stuff, but Dr. Huang has been successful in doing the dogs, which we showed in the Nature 2005 paper. He's doing pigs and cattle based on characteristics that are needed for agriculture, as well as has done some cloning of gray wolves as an example of an endangered species. Should Dr. Huang be allowed to continue receiving grant funds to pursue scientific research on cloning or potentially other topics? Or, even kind of beyond that, should Dr. Huang be able to continue efforts to clone adult organisms? Basically, if he's had misconduct in the past, how do we ensure the veracity or accuracy of the results he has going forward? So some of the references associated with this are included in the slide. Thank you and have a great day.